Today we are going to be talking about that sneaky question in the alternative to practical paper that asks you to design your own experiment. So it's all about designing an alternative experiment, sort of leveraging off the experiment that they've given you, and what would you tweak and how would you then turn this into an experiment looking at something else. So what we're going to do today is we're going to work through the steps that you need to consider when you're building your response to that kind of question. And then we're going to go look at a past paper question to see it actually work in action. So, building an alternative experiment. The thing is that what we're going to do here is we're going to bring in everything we've been talking about with regards to variables. Remember, we've touched on independent and dependent variables. We've talked about hypotheses and aims, and all of that is coming together in how you do this. These questions are often worth about 6 marks, and there are 6 things to consider. So it's a nice, easy way to remember things. Whenever you're going to set up an experiment, you want to start from the fact of, what am I looking at? So the first thing that you want to consider when you're building a response for this is, what is my independent variable? Remember, the independent variable is the one that's causing the changes. It's the one that you're setting at specific levels. It's the one that you're testing the effect of. So if they say, how would you now set up an experiment to test the effect of blah, 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 blah is in your independent variable. When it comes to building your response here, there's 2 things you want to particularly bear in mind. For your independent variable, you want to specify the range. If you can spell V, it helps. Okay, and you want to be really specific. So we don't want to just look at a range of temperatures or a range of pH values. We want to say, okay, we're going to set up this experiment. So we are going to use temperatures of this, 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 and this. Or we're going to use pH values of this, 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 and this. Okay, we don't want to be vague. We want to be specific. So you would aim for 5. So aim for 5 specific values. And the other sneaky thing to remember here, with their units. Okay, so you would want 10 degrees C, 20 degrees C, 30 degrees C, 40 degrees C, and 50 degrees C. That then gives you a specific range with specific values. So this is, of course, if you're going to look at different experiments, it could be different. Maybe you're looking at plant growth in the light and in the dark. So then you're not going to have 5 values. You're going to have light and dark. That's it, end of story. But this is the thing that you want to keep in mind. When you've got these ones which are working with more different options, then give 5 specific ones and don't forget your units. The next thing, right? Okay, this is what we're going to test. This is our sort of keystone of our experiment. But remember that when we have our independent variable, we want to be sure that the effect that we see is a result of that independent variable. So what other things do we need to control? So we want our controlled variables. Here, generally aiming for about 2 controlled variables. Your controlled variables are often going to be the same as the ones in the experiment that they've given you. So maybe you are controlling one thing that's the same, and then one thing that was changed, your independent variable in the previous experiment now becomes one of your controlled variables. Okay, so if you were testing the effect of temperature, now you want to test the effect of pH, then temperature must stay the same. So again, this is a bit more fluid here, because it's going to very much depend on your experiment. But what you want to do is you want to aim for 2 And you want to give details on how we would control them. How are you going to control temperature in your experiment? How are you going to control pH? Okay, so this is now going back to that lesson that we did all about different types of variables. So, controlled variables, how is each variable controlled? So we've got our independent variables, we've got our controlled variables. Now we're having to look at the dependent variables. What is it that we are going to measure in order to see the effect of our experiment? And how are we going to measure it? So those are our two things. What are you going to measure and how will you measure it? So now you've thought halfway through this experiment, but with a great deal of detail. So you've got your independent variable, your control variables, and your dependent variables. Then you want to think, okay, well, it's all very well to list variables and what I'm measuring and how I'm measuring it, but I need to give some sort of description of my experiment. So you're looking just for a sentence or two of a brief description of your experiment. And here, you want to consider also those things that we talked about in terms of accuracy and reliability. So when it comes to the brief description of your experiment, you're going to also think about accuracy. So the need, for example, to use 20 centimeters cubed of your independent variable. If that's a solution that you've changed concentration of, you would want to make sure you've got the same amount of it in each time. Okay, so your controlled variables, they're all sorted, and your dependent variables, what you're measuring and how you're measuring it, you've covered it there. So when we're describing and we're thinking about accuracy, it's about what are we doing to each of our variations here? And are they all the same? Because remember, when it comes to control, you've got two aspects of control. One is you controlling the situation, and the other is that you're controlling the variables that could impact on the situation. So if we're going to control the situation, we want to make sure that every test tube is exactly the same, except for the thing that we want to change. Or the environment of every plant is exactly the same, except for the one thing we want to change. Okay, so we need to keep in mind those two elements of control. So if you're a little bit confused about control in terms of controlled variables and controlling experiments, then head back into that lesson so that you can have another go through of what it is control is really meaning. So here we've got our four things, and we've kind of built our experiment. So what are those other two things that you must always consider when it comes to building an alternative experiment? We're going to fit them on here somehow. So, number 5. The thing that you want to always remember we said accuracy, and I briefly mentioned reliability. So how are you going to make your experiment reliable? You're going to repeat it. Every time you do an experiment, when you're building an alternative experiment, you're going to include repetition. And you're going to play safe, and you're going to always repeat a minimum of 5 times. So it's not going to be a case of, do I say, repeat it at least 5 times, or do I say repeat it a minimum of 5 times? No, you can tell them, I'm going to repeat my experiment 5 times. Because remember, here you're building the instructions for this experiment. You're not talking in general or in vague, or this is sort of how you would approach it. You're saying, in order to do this alternative experiment, I'm going to do this, this, this. So when it comes to repetition, I'm going to repeat it 5 times. So that would be 5 times at each temperature, or 5 times at each pH, or 5 plants in the light and 5 plants in the dark. Okay, whatever it is that we're testing, we're going to do that test 5 times. So it's a different 5 to our 5 specific values. We're now going to have 5 times at each specific value. So if we've got 5 temperatures and we're going to repeat 5 times, that's 25 experiments. But that gives us the reliability in our results that we can actually go forward with. So we have our repetition. And the last thing, this might be an alternative to practical paper with an alternative experiment in it, but the thing is that at the end of the day, these are instructions for a real-life experiment. And in a real-life experiment, you always got to consider safety. So your last thing that you're going to add in here is a safety precaution. Or a few, depending on what you're working with. Okay, so this is things like you're going to use gloves, you're going to use protective goggles, you're going to use tongs to handle hot test tubes. So things like gloves, glasses, tongs. So you've got to think through your experiment and what it is that you're doing. And then if you were standing in a lab actually running this experiment, what is it that you'd have to be so careful of in terms of safety? And you always add that in. Okay, so building an alternative experiment, you can see now we've got here to 6 points. And we often have 6 marks to achieve in our alternative experiment. But the thing is, we've got so much detail under all of these that they are often actually worth, when we write all this out, more than 6 marks. So you're really covering your bases and you're getting then a proper explanation, a proper description of an experiment that somebody could go away and work with. That's the level of detail that you're aiming for. Okay, so here we have a piece out of a question paper. We're not going to work through the whole question paper. We know we can't do that in the time we have available. Okay, so we are working here through Summer 17, paper 61, question 2, and we've jumped all the way through to a description of another experiment. So they tell us in the paper that some students measured respiration in yeast using a culture of active yeast. Yeast produces a gas during respiration. So 2 syringes were filled with 20 centimeters cubed, that's nice, we like detail, of the active yeast culture, and each syringe was placed into a large test tube containing water at 35 degrees C. Both were placed in a water bath at 35 degrees C, as shown in figure 2.2. So there is an attempt here to control the temperature. Okay, so the volume of gas in each syringe was measured every 5 minutes for 25 minutes. So if we have a look here at the picture, here at the outside we've got our beaker with our water bath at 35 degrees C. We have our big fat test tubes with water at 35 degrees C. And we have a syringe containing the active yeast culture. And at the top then is the gas produced by the yeast. So as this yeast produces gas, this is going to get bigger and bigger and bigger, more gas in here, which means it's going to force the level of the yeast down, and the yeast can just run out here into the water. So the more gas we have that is produced, the more our level of yeast is going to drop in our syringe. If we move then to look at the questions, first we want to work through these because we don't want to go straight into our description before we're sure we really fully understand what's going on in this question. So they ask us to state 2 variables that have been kept constant in this method. So what have we controlled in order to see the effect of what we're looking at? Okay, so we have controlled. Remember, we underlined it twice. We were very happy to see all this 35 degrees C. So we have kept constant the temperature. Both of these experiments are happening at exactly the same temperature. Another variable that's been kept constant, we circled it. Anytime we measure something with a volume, that is again a way of keeping things constant. So our starting volume of yeast, but it's not just yeast, it is a culture of active yeast. So it's a starting volume of active yeast culture. So now the thing is that is two options. Okay? There are other things that have been kept constant here. So if we think about it, the other thing we underlined was every 5 minutes for 25 minutes. So we have also kept constant the time intervals. Okay, both of them are measured at exactly the same time. We've also used only one culture. So we have also controlled the yeast culture used. So the question has come through about whether we would lose marks if we didn't mention the fact that it is a starting volume of, and specifically, active yeast culture. So the sort of bare minimum would be volume of yeast. But the thing is that when we're doing questions like this, what we want to do is aim for that full descriptive so that we get in the habit of building that, looking in detail at what it is we're studying. And what we're studying here is not just yeast, okay? Because just yeast is not going to do anything. You can go to your local grocery store and buy just yeast and it's sitting in a packet, it's not respiring. So the important thing here is that it is active yeast. So this is one of those additional words that I would not want to skip out on. If you're going to skip out on one, skip out on culture. It's active yeast, that's the reason why it is actually producing that gas. So in this case, I would not take that risk of missing the active, okay? Our independent variable here is our active yeast. So moving on, they ask us to identify one source of error in this method and suggest an improvement. One source of error, only one. Okay, so yes, we did underline and we were quite happy with a few things here, but this is not a very detailed description and therefore we've got open to error. So, one thing that rings in my mind straight away is whenever we're using a water bath like this, a beaker water bath, that temperature is not controlled for the duration of the experiment. If you have a cup of tea and you come back to that cup of tea 25 minutes later, is it going to be the same temperature? Not a chance. So it's the same with this water bath. Okay, so the temperature changes over the course of the experiment. Sorry, skipping letters, words. Over the course of the experiment. So the impact is the same on both test tubes because both test tubes are in this cooling down water bath, but there is an effect of temperature over time. The temperature at the end of 25 minutes is not the same as the temperature at time 0. How would we change this? We would actually want to maintain that 35 degrees C for the duration of the experiment. So that means we need our thermostatically controlled water bath. So the improvement would be use a thermostatically controlled water bath to maintain temperature throughout the experiment. Now, I mean, there are other options. I mean, these syringes that they use here, this scale is quite a broad scale, so that makes it difficult to read. And remember, any scale that is difficult to read creates a source of error. So if you used a syringe with a finer scale, that would also be an error and an improvement. Whenever you have a culture of active yeast or any kind of solution that is mixed, of course, remember, if you don't mix it before you take out your sample, you could have different concentrations. Now, they didn't tell us that the solution was mixed before removing. So we might sort of infer that one would do that because it's good practice, but because it's lacking here in the description, then it could be used as a source of error. But it's not sort of such a good idea to aim for sources of error like that. You want to rather aim for the error that is there and that's blatant. So something like your effect of temperature, because we know temperature affects all sorts of enzyme-controlled reactions. So now we really understand what's going on this one. Here is the big question. Describe how the students could use the method in 2B, that was 2B that we just talked about, to investigate the effect of pH on respiration in yeast. So we want to work through all of those things that we've just been through. I'm going to start out with our independent variable. So we would use a range of pH values. I'm going to do it in bullet points here so that you can see exactly how things would work. You can see each element that we talked about. So use a range of pH values. Now we want a minimum of 5. We go from 1 to 14, but we know things are often in the middle. So how about pH 3, pH 5, pH 7, pH 9, pH 11. So we've given 5 different ones, and we've given specific values, and we've given units. So we've covered a whole lot there, all in one thing. Right, so once he's sorted, we need to ensure that things are controlled. So we're going to be a little bit more sort of controlling from the beginning here. So ensure volume of yeast culture, nonsense, volume of active yeast culture, volume of pH solution, because we're going to make buffer solutions, and temperature of water are all controlled. Excuse me. So they use 20 centimeters cubed of active yeast. We can do the same. So 20 centimeters cubed of active yeast culture. And we're going to use a syringe for that. Let's say we're going to use 10 centimeters cubed of pH buffer solution. We're going to use another syringe for that. And then, this is a new experiment, and we know already that there was an error in the last one. So, temperature of water, we're going to use a thermostatically controlled water bath kept at 35 degrees C. So we've talked now about the things, how we're going to set this up, how we're going to be nice and accurate and as controlled as possible. What are we going to measure? Our dependent variable. So we're going to do the same thing they did. I mean, there's no reason in this case why we need to change anything. So we're going to measure the volume of gas produced every 5 minutes. We're not going to go into great detail there because we would be copying a lot of their method, and they're not going to give us a whole lot of marks for copying a previous method. So we're going to focus on the differences, things like our pH values, our pH buffer, our thermostatically controlled water bath. Okay, so how are we going to generally do this in one brief description? We're going to set up tubes of active yeast culture. We're going to add our pH buffer solution, and we're going to measure the gas produced. Remember those last two things that you're always going to add in? You're going to add in repetition and safety. So we're going to repeat experiment 5 times for each pH. Not randomly, but specifically 5 times for each pH. And then our safety, we're going to make sure that we use safety goggles and gloves when handling our pH buffer solutions. Because remember, those are going to be acidic and basic there. They're not going to be nasty, not going to be very nice solutions to be getting on our hands. So we want to be particularly careful with those. So you're working through those 6 points to remember when you are designing an alternative experiment. So make sure you've got a good handle on those, and then you're reading with enough detail and understanding that you can build your own experiment.